Hey y'all, so I will be recording the metabolism part 2 for this session, and we will be going through the individual pathways for cellular respiration, as well as going through the handout too. In biochemistry, you're going to be learning the in-depth steps, or all the steps, of all the pathways in Biochem 2. But for microbiology, we tend to focus more on the big picture, as in like what's coming in, what's coming out, how much ATP is being made. But I'll go more into detail about that later on in this session once we get into the nitty-gritty of that. So I'm going to share my screen with y'all and we'll get started. So the topics we'll be going over are glycolysis. I'm just going to classify the transition step separately, but I mean, I know the professor that I decided for, he grouped it separately, but then I knew another teacher that grouped the transition step with glycolysis, or it was the first part of the Krebs cycle as well. It just depends on your professor. So another name for the Krebs cycle is the tricarboxylic acid cycle, and then you have the citric acid cycle, same thing though. ETC stands for the electron transport chain, and then PPP is the pentose phosphate pathway. It's basically an alternative pathway to the Krebs cycle. So the bacteria do undergo glycolysis, but they may not all undergo the Krebs cycle. But I'll talk more about that. ATP yield, anaerobic respiration, oxygen requirements, and then photosynthesis in the Calvin cycle. The microbiology class that I assigned for, we didn't really spend a lot of time. We barely spent any time on photosynthesis, but I have a couple videos to show y'all that I think covers everything thoroughly, and I mean, I think there's more clarification in the book if you need it, but pretty much, like I said, for microbiology, we don't really focus on nitty-gritty of the pathways or the cycles themselves. We focus on more like what's coming in and out, and whatever's coming out, how much ATP does that produce. Okay. So the first one, we're talking about the metabolic steps for organisms. So those steps are, we do first glycolysis. Another name for glycolysis is Engen-Meyerhof. We have the Krebs cycle, like I said, PCA or CAC, and then we have the electron transport chain. Okay. So pretty much what's going on in glycolysis, it's the breakdown of glucose to produce pyruvate and energy, and that energy is in the form of ATP. So glycolysis is the central pathway for metabolism. And like I just said, it's basically the breakdown of glucose to produce pyruvate. That's usually how you abbreviate pyruvate, but you spell it like this: Pyruvate and ATP. Okay. So the pathway or process of glycolysis oxidizes glucose, which is a derivative pyruvate, glycolysis is seen in many organisms, not just in humans, from microbes, humans. This is a major point. It occurs anaerobically or aerobically. Anaerobically or aerobically. So basically, oxygen is not required for glycolysis. Okay. Basically, what we're saying can occur anaerobically or aerobically. Basically, the organism can still undergo glycolysis and function with or without the cell can function with or without oxygen whenever glycolysis needs to be taking place. And glycolysis requires 2 ATP to prime the glucose. I'll point that out to you in the handout. So basically, you make a total of 4 ATP in glycolysis, but you have to use 2 ATP in the process to make the total 4 ATP. So the gross number of ATP for glycolysis is 4, but the net gain is 2 ATP because you had to use 2 ATP in the process of glycolysis. Yes, oxygen is not required. I already said that. And substrate level phosphorylation occurs in glycolysis and in the Krebs cycle. Okay, oxidative phosphorylation is what takes place at the electron transport chain. This is important. Okay. But with substrate level phosphorylation, you produce little ATP. Produce specifically a net gain of 2 ATP. But substrate level phosphorylation is, I'm just going to write out a brief definition. It's the process of forming ATP by the physical addition of a phosphate group to ADP. Or another way to say it is substrate phosphorylation is the synthesis of ATP by the direct transfer of a phosphate group from a substrate to a molecule of ATP. Basically, you're just sticking on a phosphate group from a substrate to ADP. That's what's happening in substrate level phosphorylation. And the direct transfer of a group from a substrate. The substrate is a high energy intermediate to a molecule of ADP. Okay. So I'm just going to draw the example of substrate level phosphorylation, and that would be going from HEP to pyruvate. So I'm just going to do P with a circle to indicate the phosphate for simplicity purposes. Okay, so here's a substrate. HEP. And this is the ADP, adenosine diphosphate. So phosphate, okay? Then pretty much you just take this phosphate and add it to the group over here, and you have pyruvate left over as your product. Pyruvate has 3 carbons, so we have 1, 2, 3, 1, 2, 3 carbons, and we have ATP, adenosine triphosphate, and that makes sense because we have 3 phosphates now. Yeah. So substrate PEP, ADP, then the product is pyruvate, and then ATP. Okay. Now I'm going to move on to the transition step. So basically what happens with the pyruvate before the Krebs cycle takes place. So another name for the transition step is the linker reaction. In prokaryotes, oxidation occurs in the cytoplasm. So glycolysis took place in the cytoplasm, right? And this transition step is occurring in the cytoplasm as well. Occurs in mitochondrial matrix. And pretty much in the transition reaction, you generate NADH and acetyl-CoA, which is a precursor metabolite. And I will define that just a sec. So precursor metabolite is basically an intermediate molecule in catabolic and anabolic pathways. Here, I'll finish writing. I'll just write it out. Intermediate molecule in catabolic or anabolic pathways that can be oxidized to make ATP or be used for biosynthesis. Okay. Anyway, so this is the you also generate CO2. So you generate NADH, CO2, and acetyl-CoA. So we have pyruvate here, right? Pyruvate coming in. You have NAD, NADH, and then CO2 also leaves because you end up having coenzyme A coming in. So with acetyl-CoA, you have 2 carbons instead of 3 because we lost 1 here. And then with this acetyl-CoA, basically the acetyl group is transferred to initiate a round of the Krebs cycle. So all this needs to take place before the Krebs cycle. Okay. Okay, now I'm going to move on to the Krebs cycle. And basically the Krebs cycle is a sequence of chemical reactions. I'm just abbreviating reactions in which molecules are broken down in the presence of oxygen to generate energy. And that energy is in the form of ATP. It's also used aerobically and anaerobically. For each molecule of glucose, you obtain 2 pyruvate. So that transition step that I showed you with one set of pyruvate, those 3 carbons, that's 1 pyruvate. Technically, this is happening twice. Okay, so since you obtain 2 pyruvate, you're going to have 2 acetyl-CoA going into the Krebs cycle, right? So if you have 2 acetyl-CoA and only 1 acetyl-CoA sets off 1 round of the cycle, then technically you're going to be doing the Krebs cycle twice. So cycle twice. So you obtain, I won't go into detail about the GTP, but basically you obtain 2 ATP, you obtain NADH and FADH2. So the NADH and FADH2 carry electrons that are used to generate ATP at the electron transport chain, at the membrane. So you get this to go from 2 GTP to 2 ATP. The energy released when coenzyme A is removed is harvested as GTP and is harvested as GTP and then converted to ATP. Yes. Okay. Yes, I'll go ahead and show the first video before I continue, and I think this will make more sense with this.
Humans consume food for energy and use oxygen for respiration in a process referred to as metabolism. Believe it or not, bacterial cells carry out almost exactly the same processes. Understanding bacterial metabolism can be difficult. There are many pathways and enzymes, and it is not always clear how they link together. In this lesson, we will use a simple way to visualize the metabolic pathways. So stick with me, and by the end, you should have a good understanding of some basic pathways and how they fit together in a bacterial cell. One of the easiest ways to visualize the connections between metabolic pathways is to imagine the various pathways and processes as individual blocks. Let's use these to build a metabolic model of a bacterial cell growing by aerobic respiration. Aerobic respiration is the process in which a compound is oxidized using oxygen as the terminal electron acceptor. That definition probably doesn't make much sense yet, but by the end of the lesson, it will come together. Okay, let's start building. Our cell will use glucose as an energy source under oxygen conditions. The first metabolic block we will choose is glycolysis. The glucose is taken up by the cell and broken down via glycolysis. If you dissect the term glycolysis, glyco means sugar, and lysis means to split. So, very simply, glycolysis uses enzymes to split glucose molecules into pyruvate. During the breakdown of glucose, electrons are liberated and used to form NADH, or nicotinamide adenine dinucleotide, an important electron carrier in the cell. Let's take a quick time out. In biological systems, energy is captured and conserved using oxidation reduction reactions, or redox reactions for short. Basically, redox reactions involve the moving around of electrons between molecules, with oxidation referring to the removal of electrons and reduction referring to the addition of electrons. So, to relate that to glycolysis, the glucose is oxidized, meaning the electrons are stripped from it, breaking it down into pyruvate. The pyruvate formed by glycolysis serves as the connection between the glycolysis block and our next block, the citric acid cycle. The citric acid cycle is a series of enzymes used to break down pyruvate all the way to carbon dioxide. This cycle oxidizes the pyruvate, capturing its electrons and depositing them on NAD to form NADH. The NADH formed during glycolysis and the citric acid cycle connects to the next block in our pathway, the electron transport chain. The electron transport chain is a series of enzymes, proteins, and molecules embedded in the cell membrane that pass along electrons. The components act as a sort of conveyor belt for electrons. The first enzyme complex is a dehydrogenase that removes the electrons from NADH and passes them to a molecule called quinone. The quinone molecules pass them on to a series of proteins called cytochromes that then transport them to the final multi-cytochrome enzyme in the chain called the terminal oxidase. The terminal oxidase is an enzyme complex that transfers the electrons to oxygen, generating water and terminating the electron transfer process. The most important part is that as the conveyor belt passes along the electrons, energy can be captured to simultaneously pump protons out of the cell. This results in a gradient of protons across the cell membrane, high on the outside and low on the inside. This proton gradient feeds the next block in our metabolic model, the ATP synthase. Okay, let's use one more timeout. The best analogy for the proton gradient is the electrical current you use to power the appliances in your house. That current is simply the flow of charged particles, electrons, from a source, the outlet, to a refrigerator, the appliance. The proton gradient is referred to as the proton motive force, and like the current you access through your outlets, results in the flow of charged particles, in this case protons. The proton motive force is an energized state across the membrane due to the proton gradient. Just like the electricity in our homes, this energized state can be measured in volts. The ATP synthase utilizes the proton motive force to generate ATP. ATP, or adenosine triphosphate, is the energy currency of the cell. It is a high-energy molecule that is used to build all of the cell components and structures. The ATP synthase is located in the cell membrane and is able to open up a channel that allows the protons to flow down the gradient. The energy provided by the flow of protons results in a force that spins the ATP synthase. That spin energy is used to catalyze the addition of a phosphate group to ADP, or adenosine diphosphate, to generate ATP. This process of ATP synthesis is referred to as oxidative phosphorylation, or the generation of ATP using a proton motive force generated by an electron transport chain. Now we have completed our metabolic model cell. This cell is using glucose as a source of energy and oxygen as an electron acceptor to generate ATP. Now, we have done a great job building a model cell, but rarely is a cell growing on pure glucose. Out there in the real world, there's a vast array of potential growth substrates, and we would have many additional blocks to choose from to build a functioning bacterial cell. Some of these blocks would completely replace one of the blocks we use today. Other blocks would fit neatly into our existing pathway, and still other blocks would replace almost every block in our original model. Aerobic respiration is the process in which a compound is oxidized using oxygen as a terminal electron acceptor. In the beginning, this may have seemed complicated, but now we can use our model cell to better understand that definition. At the start of the process, glucose is oxidized. Through a series of steps and pathways, the electrons that originated from glucose end up in the electron transport chain. The electron transport chain conveyor belt passes the electrons along while simultaneously pumping protons out of the cell. At the end of the chain, oxygen is reduced to water, and the electron's work is done. Importantly, the whole goal is to generate the proton gradient that results in a proton motive force that powers the ATP synthase to generate energy for the cell, a process termed oxidative phosphorylation.
Okay, so I know that was a bit of a long video, but I thought it covered everything that your handout covers in terms of cellular respiration. I mean, I know it doesn't mention the alternative pathways, but anyway, so I thought it was a good overview just to let you know, like, where what's going where, and what oxidative phosphorylation is. So, anyway. Okay. So we talked about that. Now, actually, before I get into the ATP yield, I'll go ahead and go over the handout. So, beginning of your handout, it's a brief review over like what's reduced, what's oxidized, reducing agent, oxidizing agent. Okay, I briefly talked about that last session, and then glycolysis. So another name for glycolysis is Enden-Meyer-Hof, but there's a step in glycolysis where glucose 6-phosphate is converted to pyruvate. Technically, that step is called Enden-Meyer-Hof harness. Anyway, so I wrote out these bullet points about oxidized glucose / derivative to pyruvate, produces a little ATP through substrate level phosphorylation. Wrote that out. I told you what substrate level phosphorylation is, oxygen not required. Pretty much the more stars next to the bullet point, the more important it is for you to know. And so there are more steps in glycolysis. The professor that I assigned for, these are the steps that he wanted to highlight on because he just wanted to know, like, okay, so in this case, glucose 6-phosphate, okay, like this is an important step because glucose was phosphorylated. So, and how is the glucose phosphorylated? By using ATP. So, and that the glucose is phosphorylated through group translocation, and that's what I talked about in the last, like near the end of the last session. So anyway, then fructose 6-phosphate to fructose bisphosphate, use ATP to phosphorylate fructose 6-phosphate enzyme is phosphofructokinase, which is the rate determining step of glycolysis. Like I said in biochem, for each step you're gonna be learning the individual enzymes and like which group is going where. You don't really need to worry about that for micro per se. If you do want to know, I mean honestly, you could probably just Google chemical structures and glycolysis, and I'm sure a world of information will come up. So the point is, from the first part of glycolysis, you've used up 2 ATP in order to phosphorylate these compounds, okay? Then DHAP dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate, and then oxidation of glyceraldehyde 3-phosphate to bisphosphoglycerate to 3-phosphoglycerate, yeah, and then phosphoenol pyruvate. Okay, so the professor wanted to highlight these steps to say, okay, this is the part where the pathway goes in parallel, this is the part where substrate level phosphorylation occurs, and this is how much ATP is made. So basically, out of what's going into glycolysis is glucose, and what's coming out, what's going into glycolysis is glucose, and then you end up using up 2 ATP, and then what's coming out are a total of 4 like the gross total is 4 made, but out of that 4, you had to use 2 in the pathway of glycolysis. So technically, you really only gained 2 ATP from glycolysis. So that's why I said earlier, substrate level phosphorylation produces very little net ATP because they only produces 2 ATP. Then transition step, link reaction involves oxidation of pyruvate. Okay, it generates NADH and precursor metabolite, acetyl-CoA. I wrote that out. Yes. Tricarboxylic acid, Krebs cycle, amphibolic. Okay, so amphi means both, so it could be either anabolic. I mean, it can be both anabolic and catabolic. And then under aerobic conditions, the cycle produces free energy or reducing power as NADH and FADH2. The final electron acceptor for when it's occurring aerobically is oxygen, but anaerobically, okay, anaerobically the final electron acceptors are different because there are alternative electron carriers, and those different electron acceptors are nitrate and sulfate, not oxygen. But for fermentation, the final electron acceptor is an organic molecule. So it's not even nitrate, sulfate, or oxygen. It's an organic molecule. Then I talked about the cycle turning twice. This means succinate to fumarate. And generate. So this is just talking about the metabolites and Krebs cycle. These are like cherry picking the important details from these pathways. Depending on who your professor is, they should emphasize in lecture like how detailed he or she wants you to know about these pathways for microbiology. Okay, but I just know for the class that I assigned for micro, they really need to know more of like what was coming in and out for each pathway, and then if you started off with a certain number of glucose, how many ATP would be made after the electron transport chain. And then alternate pathways. I'll be talking about the pentose phosphate pathway in just a minute. But basically, these alternative pathways are pathways that can either, like the pentose phosphate pathway, they go through, glucose would go through glycolysis, and then for pentose phosphate pathway, it'd go glycolysis, then pentose phosphate pathway, and it would skip over Krebs. So it just depends if this respiration is anaerobic or aerobic respiration. Then, let's see. Oh, perfect. I was going to write this out, but it's already here. So respiration uses NADH and FADH2 to synthesize ATP. That's true because NADH and FADH2 are electron carriers. They carry the electrons to the electron transport chain, drop it off, and then the electron transport chain generates a proton motive force, which ATP synthase uses that proton motive force in order to phosphorylate an ADP to make ATP. Yes, oxidative phosphorylation couples electron transport and ATP synthesis. Chemiosmotic theory. The chemiosmotic theory is that we talked about that with oxidative phosphorylation, and then occurs in the cytoplasmic membrane. Okay, it's all right here, honestly. Like I don't really have anything extra to explain to y'all because it's right here on the handout. I recommend that if you need more visuals, there are plenty of videos on YouTube or the book if you want to write out something like on your study sheet or anything like that. I just didn't draw anything because I would have drawn the whole schematic, and I know for micro we didn't really need to know that as well. You really just need to know that more for biochem too. Then electron transport. Redox reactions occur in cytoplasmic membrane. Yes. for prokaryotes, and then ejects the hydrogen atom outside of the cell or between membranes, creates proton gradient, proton motive force used by cells to make ATP. Yes, because you have a proton motive force because of that separation charge. The plus charge is outside the cell, and then the negative charge is inside the cell and the cytoplasm. The hydrogen atoms flow back into the cell via transport proteins. Energy is used to make ATP by ATP synthase. Yes. So, okay, hold on. Then ATP yield. Okay. This is talking about what's coming out of the individual pathways. So glycolysis, you make 2 NADH, which are equivalent. 1 NADH is worth approximately, like if you round up, it's worth approximately 3 ATP. So that's why you have 6 ATP made here. And then transition step 2 NADH because you have 2 pyruvate. So then you that's how you get 2 NADH. So you get 6 ATP, and then the tricarboxylic acid cycle, you make this much. So 1 NADH, if you round up, is worth approximately 3 ATP, and then 1 FADH2, if you round up, is approximately worth 2 ATP. So 6 times 3 is 18, 2 times 2 is 4. And so that's where we get the ATP. This is how much ATP we get from the energy carriers. But we also made a little bit of ATP from the substrate level phosphorylation and glycolysis, right? Remember, we made a gross of 4 ATP, but since we had to use 2 earlier on in glycolysis, we technically had a net gain of 2 ATP. And then tricarboxylic acid cycle, substrate level phosphorylation also occurred there, and that's how we got our 2 ATP there. So in prokaryotes, the maximum the theoretical yield for prokaryotes is 38 ATP per mole of glucose. Okay, I already talked about that last session. Our class didn't really talk about fermentation because y'all should have learned that already in Bio 1 and 2. So I don't really have anything to add for fermentation other than like what's here on the handout. And I do have a video to show y'all for photosynthesis. But I have a few notes for we already talked about respiration with the electron transport chain, and I have a couple notes for the pentose phosphate pathway, and I just wanted to rewrite out the ATP yield for that. Okay. Alright. So the bottom line is that these individual pathways create energy. Okay. It's like the big open picture. The PPP, pentose phosphate pathway. Or you can write it as glucose 6-phosphate. This is just some extra information for the pentose phosphate pathway if you need to know. But I'll be honest, this metabolism handout is your best friend for the metabolism section because this handout overlaps really well with the lecture material. So, in my personal opinion, if you know this handout well, you should be fine for micro. And it's kind of like a Cliff Notes version to what you'll be learning in Biochem 2. So these three use the Entner Deuteros. Make sure I spell it right. Deuteros pathway. And this pathway links the pentose phosphate pathway to triose phosphate and pyruvate. I doubt you need to know this part in detail, but you never know, depending on your professor. Okay, so just to be clear, the ATP yield for aerobic metabolism, the theoretical yield should be approximately 38 ATP per 1 whole glucose. Okay. So remember, substrate level phosphorylation occurs at glycolysis and the Krebs cycle. Oxidative phosphorylation occurs at the electron transport chain. But where are all these electrons coming from? Those electrons are coming from the energy carriers that came from glycolysis, the transition reaction, and Krebs cycle. I know I'm kind of rewriting this portion from your handout, but if you write it out, it'll help you commit it to memory better. And just a side note, whenever I do the equal signs like this, I'm sure for math you remember that means approximately, but just in case. Technically it's more of a decimal, like it's 2.5, I think, 2.6, I think it's 2.5. But anyway, I'm just rounding it up because that's the professor that I assigned for, the professor rounded up like this. Okay. Transition. So this is a total of 34 ATP here, just from oxidative phosphorylation. So for sure, the majority of ATP is made at the electron transport chain through oxidative phosphorylation. So the total from substrate level phosphorylation is 4. So there is like a night and day difference in the amount of ATP that's made in substrate level phosphorylation versus oxidative phosphorylation. So if you combine these two, the total max for prokaryotes is 38 ATP. So, hold on. The total for prokaryotes is 38 ATP. There you go. Okay, I just have a couple more things to go through, and then we'll be done. So we'll talk about the bottom line, anaerobic respiration. There is more about this in the Nestor book, but we primarily focused on aerobic respiration. But it's still good to know, especially like if you're going for marine biology or ecology, something like that. But aerobic respiration produces more ATP than anaerobic respiration. And when I was talking about the electron acceptors earlier from the handout, the final electron acceptor for aerobic respiration is oxygen. Final electron acceptor for fermentation is an organic molecule, and then final electron acceptor for anaerobic respiration is what I'm fixing to write here. Either they're oxidized an organic molecule, such as nitrate, sulfate, CO2, or facultative obligate anaerobes. So you're like, oh, what are facultative or obligate anaerobes? Good question. I'm going to talk about that now. I have an oxygen requirements table that I recommend that y'all know. Okay. So obligate aerobe, facultative anaerobe, obligate anaerobe, microaerophile. This is microaerophile, and then aerotolerant. Aerotolerant anaerobe. I'm saying out loud just in case you can't read it. Okay, so these are test files or test tubes, and the dot come drawing is the bacterial growth. Okay. Okay. So for an obligate aerobe requires oxygen for respiration. So where is the closest source of oxygen for this tube? It's near the surface. So there's all the bacterial growth is going to be near the surface. Bacterial growth. Facultative anaerobe, there is like a high concentration near the top, but there's also a good number spread throughout the tube. Oh, I forgot. Grows only when oxygen available. A dead giveaway is the aerobe, obligate aerobe. You can think like obligate as an obligation. So this aerobe is obligated to grow in the presence of oxygen. So the examples here are algae, Indius, and Arcuria rosopula. Okay. Then facultative anaerobe is a bit different because I don't really have like a catchy thing to remember this, but for anaerobe, you're thinking, okay, so it can grow when oxygen is not present, but you see here that there is bacterial growth throughout the whole tube, even near the surface. So facultative anaerobes can grow in the presence of oxygen or not in the presence of oxygen. So basically, facultative anaerobes uses oxygen for respiration if the oxygen is available. Okay. So examples of this could E. coli and the Vibrio genus of the subspecies. Now we have obligate anaerobes. So think, like I said, all your aerobes are obligated to grow in the presence of oxygen, or to grow, or to use, yes, grows only when oxygen is available. Well, obligate anaerobes, those anaerobes are obligated to grow when oxygen is not available. So this would be about like less than 5% oxygen. If not oxygen. Example, so the oxygen would be here, like near the surface, right? So the obligate anaerobes want to grow as far away from that surface where that oxygen is as possible. Okay. Then we have microaerophile, micro meaning small, aero meaning like air, oxygen. And the suffix phile means to like or to love. And so that's this word saying like this bacteria likes to grow in small or little bits of air. So it does require oxygen for respiration, but it doesn't need a whole lot of oxygen for respiration. It doesn't need a lot of oxygen to grow, okay? So this could be like 5 to 10% oxygen or 5 to 10% carbon dioxide. So 5 to 10% carbon dioxide, if the bacteria grew in the presence of carbon dioxide, then that means it would be a capnophile, not a microaerophile. But I digress. Anyway, so what I'm saying is that this bacteria, the microaerophiles, can grow in the presence of a small percentage of oxygen or a small percentage of CO2. Okay. And these examples are Campylobacter and Neisseria and Spirillum Okay, so with aerotolerant, Spirillum was the last example here. Spirillum, yes. And then aerotolerant, basically aerotolerant anaerobes that tolerates oxygen pretty much. So bacteria can grow anywhere in the tube. It's not really concentrated in one area. It grows equally well with or without oxygen. And the examples are Enterococcus and strep, like streptococcus, and then Pyogenes. So there's that table. Yay. This should help you with the oxygen requirements. I know that a lot of questions on the test for micro, it's more of like, oh, which of the above are facultative anaerobes, or recibo microaerophiles. Like I said before, a lot of microbiology is categorizing things, knowing how things are connected. It's not so much a concept, it's more of being able to associate or connect these different topics into this specific category. Okay, so just keep that in mind while you're studying. Okay, we saw fermentation on the handout, and now we pretty much have photosynthesis left. And I have a couple notes to put onto that. Oh, wait, actually, I think it's on your handout. I wanted to say. Yes, okay, here it is. I don't need to write this out. Occurs in plant chloroplasts, algae, and some bacteria. Goal is to fix carbon. Remember, I said whenever you're fixing an element, it's like you're converting it from inorganic to organic. You're converting an inorganic substance to organic substance. Two stages in photosynthesis, the light and dark reaction. Pretty much you have the light reaction, and then sometimes people don't say dark reaction, they may just say Calvin cycle. You have cyclic and non-cyclic phosphorylation, and then you have Calvin cycle. Okay, I have two videos to show you for this. These, again, like what I said about the specifics that are shown here in this handout, it just depends on what your instructor wants, okay? Just keep that in mind. But I think these two videos covered this part well, and it'll save time. Oh yes, I also wanted to show you all this. This should look familiar from Bio 1 and 2. This just shows you the light reaction, which photosystem we're talking about, and just the differences here, what's leaving and what's being integrated into the Calvin cycle, what's leaving the Calvin cycle and being integrated into the light reaction. Okay, so I think that's a very helpful diagram. And we'll watch the photosynthesis one first. Oh wait, I think I already have that up. Oh no, I didn't. Sorry. So I'll watch this, and then we'll do the Calvin cycle, and that should wrap up this video.
In order for plants to grow, they need inputs of carbon dioxide, water, and energy. The chemical process by which plants use these resources to manufacture glucose, the building blocks of plants, is called photosynthesis. In the process, oxygen gas is produced as a byproduct. The energy for photosynthesis originates in the sun and arrives at the earth as sunlight. This light has both a wave and a particle nature. The particles, or photons, are the smallest units of light. Photons oscillate along a path, which is measured as wavelengths. The light emitted from the sun contains photons in a wide spectrum of wavelengths, called the electromagnetic spectrum. Photosynthetic organisms use only a small portion of the electromagnetic spectrum, called visible light. Photosynthetic organisms contain pigments that facilitate the capture of wavelengths of light in the visible light range. The color of the pigment comes from the wavelengths of light reflected. Plants appear green because they reflect yellow and green wavelengths of light. Red and blue wavelengths of light are absorbed by these pigments and provide the energy that is used for photosynthesis. Within eukaryotic photosynthetic organisms, also known as photoautotrophs, the chemical reactions of photosynthesis occur within plant cells in specialized structures known as chloroplasts. Photosynthesis consists of two sets of reactions, the light-dependent reactions and the Calvin cycle.
Okay, I know she said eukaryotic cells, but I've watched this part, and this does apply here for micro.
Within the chloroplast are small disc-like structures called thylakoids, which are surrounded by a fluid-filled space called the stroma. The reactions that synthesize glucose, the Calvin cycle, occur in the stroma. The light-dependent reactions occur in the thylakoid. It is here that conversion of light energy to chemical energy is initiated. In most photosynthetic organisms,
okay, I wanted to point out this. This is the graph there. So this is just to show the overview. Photosystem 2, light-dependent reaction, and the stroma here at the Calvin cycle. It's kind of that other picture I showed y'all a minute ago.
Thylakoid. It is here that conversion of light energy to chemical energy is initiated. In most photosynthetic organisms, thylakoids contain pairs of photosystems, called photosystem 1 and photosystem 2, that work in tandem to produce the energy that will later be used in the stroma to manufacture sugars. The photosystems of the thylakoid consist of a network of accessory pigment molecules and chlorophyll, the molecules that absorb the photons of light. Within the pigment molecules,
So this just shows you, like whenever I was talking, or whenever I mentioned I believe it's the cyclic phosphorylation, this is what it's referring to here.
absorbed light energy excites electrons to a higher state. Photosystems will channel the excitation energy gathered by the pigment molecules to a reaction center chlorophyll molecule, which will then pass the electrons to a series of proteins located on the thylakoid membrane. Photons of light strike photosystems 1 and 2 simultaneously. We will examine what happens with the
Okay. So this is being recorded. You could probably pause it at this point if you want to read these points here. I know the class didn't need to know this in detail at all, but just in case if you wanted to learn more.
The photons striking photosystem 2 first. The energized electrons are passed from the reaction center of photosystem 2 to an electron transport chain. The electrons lost by photosystem 2 are replaced by a process called photolysis, which involves the oxidation of a water molecule producing free electrons and oxygen gas.
You don't really need to worry about the photolysis part.
While this oxygen gas is a byproduct of photosynthesis, it is an important input to the cellular respiration pathways. As electrons pass through the electron transport chain, the energy from the electron is used to pump hydrogen ions from the stroma to the thylakoid, creating a concentration gradient. This gradient powers a protein called ATP synthase, which phosphorylates ADP to form ATP. The low-energy electrons leaving photosystem 2 are shuttled to photosystem 1. Within photosystem 1, low-energy electrons are re-energized and are passed through an electron transport chain where they are used to reduce the electron carrier NADP+ to NADPH.
So notice in photosynthesis, the electron carries NADP+, not NAD+. So just think NADP, the P for photosynthesis.
When the chloroplast is receiving a steady supply of photons, NADPH and ATP molecules are rapidly being provided to the metabolic pathways in the stroma. Therefore, the ATP and NADPH formed during the light-dependent reactions are used in the stroma to fuel the Calvin cycle reactions. The Calvin cycle consists of
Okay, like I said, it depends on your instructor of how much they want you to know. This just looks more like the stuff you would study more in Bio 1 and 2. For the class that I assigned for micro, they did not need to know this in detail. Like, they really just needed to know what was on the handout. And I'm not trying to, like, pigeonhole you just to the handout. That's why I'm giving you this whole video. But I'm just letting you know that you may not need to know everything quite in detail. Sometimes you just need to understand the broad picture in order to understand the nitty-gritty details, but you just have to double check with your professor on what they want. Okay.
The series of reactions that reduce carbon dioxide to produce the carbohydrate glyceraldehyde 3-phosphate.
Oh, okay. I already talked about reduction.
The cycle consists of 3 steps, the first of which is carbon fixation.
And your carbon fixation conversion of carbon dioxide gas into a solid compound. Inorganic carbon to organic carbon.
Step, carbon dioxide is attached to ribulose 1,5-bisphosphate, resulting in a 6-carbon molecule that splits into 2 3-carbon molecules. The second step is a sequence of reactions using electrons from NADPH and some of the ATP to reduce carbon dioxide. In the final step, ribulose 1,5-bisphosphate is regenerated. For every 3 turns of the cycle, 5 molecules of glyceraldehyde 3-phosphate are used to reform 3 molecules of ribulose 1,5-bisphosphate. The remaining glyceraldehyde 3-phosphate is then used to make glucose, fatty acids, or glycerol. It takes 2 molecules of glyceraldehyde 3-phosphate to make 1 molecule of glucose phosphate. Thus, the Calvin cycle has to run 6 times to produce 1 molecule of glucose. These molecules can remove their phosphate and add fructose to form sucrose, the molecule plants use to transport carbohydrates throughout their system. Glucose phosphate is also the starting molecule for the synthesis of starch and cellulose. Plants produce sugars to use as storage molecules and structural components for their own benefit. By utilizing the energy of the sun, along with inputs of water and carbon dioxide, plants act as glucose factories. Photosynthetic organisms are the primary producers of glucose on the planet. They also produce oxygen gas as a byproduct, and thus serve as the foundation of life, providing food and oxygen for the complex food webs on both land and in the oceans.
Okay. Now the Calvin cycle. Let me show you your handout. Converts CO2 to organic carbon, requires 18 ATP and 12 NADPH, features Rubisco, which is fixed CO2. Rubisco is an enzyme, and the Calvin cycle is involved in the pentose phosphate pathway. And yes, it already talked about that. Okay, good. We're just going to watch this last video, and I think that pretty much covers photosynthesis and Calvin cycle.
The Calvin cycle is a series of reactions that results in conversion of carbon dioxide into the organic molecules needed to build new cells. It occurs in the stroma, which is the area of the chloroplast surrounding the thylakoid membrane. During the Calvin cycle, carbon dioxide, CO2, is added to a 5-carbon molecule called RUBP. The resulting 6-carbon molecule is unstable and quickly splits into 2 3-carbon molecules called 3-phosphoglycerate. Using energy from ATP and reducing power from NADPH, which are products of the light reactions, the pair of 3-phosphoglycerates move through a series of reactions and are converted into 2 molecules of glyceraldehyde 3-phosphate. When several of these glyceraldehyde 3-phosphate molecules have been produced, some combine to make glucose, while others are reused in the Calvin cycle. To generate an entire new glucose molecule, the cycle has to turn several times, because each turn of the cycle adds only 1 carbon atom from each molecule of carbon dioxide.
this one, since the cyclic and non-cyclic photophosphorylation is mentioned. I think this would be helpful for y'all too. I showed this last semester too.
In bacterial photosynthesis, a single photosystem is involved. When an electron is energized by absorption of light, it is ejected from the photosystem reaction center. The electron then passes down through an electron transport system and finally back to the reaction center. The energy released during this electron transport is used to produce ATP. Since the excited electron returns to the reaction center, this mechanism for making ATP is called cyclic photophosphorylation. No reducing power needed for biosynthesis is generated in this process. Plants and cyanobacteria utilize two photosystems which work sequentially to produce both energy and reducing power. First, a photon of light ejects a high-energy electron from photosystem 2. The electron lost from photosystem 2 does not return to photosystem 2, but is replaced by an electron generated from the enzymatic splitting of water and the release of oxygen. The electron then travels from the excited reaction center of photosystem 2 down an electron transport chain and finally to the reaction center of photosystem 1. This electron transport system generates a proton motive force that is used to produce ATP. Since the excited electron does not return to photosystem 2, this mechanism for making ATP is called non-cyclic photophosphorylation. When photosystem 1 absorbs a photon of light, it ejects a high-energy electron, which is used to drive the formation of reducing power in the form of NADPH. The ejected electron is replaced by an electron from photosystem 2.
Okay. So that pretty much covers this session, and for the next one I'll be talking about bacterial growth, but we covered a lot of ground in this session, and like I said, just ask your professor on the specifics about photosynthesis, the Calvin cycle, and if y'all need to know all the steps in each metabolic pathway, or if they're going to highlight the specific steps they want you to know for the test. It just, like I said, it just depends on the professor. But as long as you understand the broad scope, as in like what's coming in, what's coming out, and what happens with those energy carriers, then you're already on the right track to understanding what's going on and why it's happening. So anyway, I hope you have a good rest of the day, and good luck studying. Bye.